Hi everyone, and I welcome all the students and participants to this live TA session of Environmental Biotechnology. So, today is Saturday, and at every Saturday 3 p.m., we have this discussion live platform where we discuss the NPTEL recommended questions and sets and answers, practice problems for the course Environmental Biotechnology. Okay, and my name is Ashutosh Behra, and I'm a PhD PMRF scholar from IIT Kanpur. I, I along with two other PhD scholars are TA for this course. So you can attend my live class on Saturday 3 p.m. every 3 p.m. Though we are reaching towards the end of this course. So today is week 11 for this live TA session, and we will be dealing with all the lectures that have been taught in the week 11. So today we'll be dealing with week 11 lectures and practice problem set from all the questions related to these lectures, where Professor Pinaki sir, who is the instructor of this course, is talking about uh, microbial oil remediation and nitrogen fixation, waste removal, phosphate removals. So these are the topics we'll be dealing today, and what I have prepared is a 10 question long multiple choice question repertoire of 10 questions, which is similar to what you will be facing in your assignments or in your quizzes and in the exam. Okay, so these questions will be of the same style, and we will be dealing with lecture week 11. I hope you have gone through your lectures and studied through it. If not, then this is the time that you can ask questions. You can We can together help solve these questions and get a hand and grasp of how questions appear in the NPTEL examination. Okay, so this is very similar to what your assignment looks like, but it's exactly not the assignment. So, general set of instructions for everyone who is joining this live class. Please turn off your mic and video chat through this presentation. Let me be the only one who speaks throughout this presentation while I'll be presenting my slides on the screen for you guys to follow. So I have prepared 10 multiple choice questions with one answer correct. You can interact with me via the chat box. So I will pose a question, you can tell me what is the correct answer, post your answers in the chat box, and then we can have a communication. So that is the form of communication we can have. And also, for your information, this is a recorded class. All the questions and doubts that I'll be solving in this live class will also be recorded and be uploaded on YouTube, the link of which will be given to you by the NPTEL authorities. Also, the material or the presentation that I have prepared for you guys will also be made available to you. So, but I definitely encourage all of you to attend these live classes and get your friends along with it so that you can make the most out of this interaction. NPTEL has asked us PhD scholars to help you understand this topic better, and we, in a collective way, are trying to make live classes for this course, Environmental Biotechnology. Okay, with that much of an introduction, my name is Ashutosh. Let us start this session. So, what I'll do is I'll try to share my screen and ask you for confirmation every time when we change the question, change the slides, or if you have any doubt or you want me to revisit the concept, then I'll be happy to do so. Okay, let us begin this session. I will start by sharing my screen. Sorry. I hope you guys can see my slides and also the slides changing. Please let me know with a yes or no. Yes, thank you. So once again, I welcome you guys to NPTEL live TA session for environmental biotechnology. So this course is taught by instructor Professor Pinaki Sir, who is at IIT Kharagpur, and my name is Ashutosh. I'm a PhD PMRF scholar at IIT Kanpur, who is helping as a teaching assistant in this course. So what I have arranged next is 10 series of questions, multiple choice questions, and this is the format that I have designed the presentation in. I'll post the question, you tell me what is the correct answer, and then I will reveal what is the actual correct answer, and then we'll understand briefly, very briefly, the concepts behind why the correct answer is the correct answer. Okay, we'll understand the concept and we will rule out other options, and thus in the process, even if you haven't got the time to revise your lectures from the recorded lectures of the NPTL, this will serve as a live revision for all your topics and concepts along with solving some of the example questions. Okay, so this is the format that we are going to follow, and this is question number 1 that I'm presenting on the screen. Biohydrometallurgy is most extensively used for which of the following metal recovery or mining? Understood the question? Biohydrometallurgy most extensively used for which of the following metal recovery? Option A, copper. Option B, gold. Option C, uranium. Option D, iron. Or option E, coal. So these are the 5 options. Only one option is the correct one. Post your answers in the chat box what you think is the correct answer, and also those who will be watching this recorded lecture, please post your answers in the comment box. Let's take a minute or two to collect some of the answers by all the participants, and then I will reveal what is the correct answer to question number 1. Okay, I will visit this question in a minute. Think about it. It's an easy question. Biohydrometallurgy most extensively used for which metal recovery? Okay, I think the time is up. Let us check if people have answered in the comment or the chat box. Ankita has answered option A. Okay. Let us find out what is the correct answer to this question. So, biohydrometallurgy is most extensively used for metal recovery of copper metal. Correct answer, Ankita. Question number 1, option A is the correct answer. So, copper recovery is the process in which hydrometallurgy is extensively used. Let us see let us understand what is biohydrometallurgy and how copper is recovered. Okay. Biohydrometallurgy is the process that uses microbes like bacteria, fungi, and plants, mostly bacteria in our context, to extract and recover metals from minerals and waste materials through biological reactions. So, if you understand, so from any waste material or biological, inorganic, or organic matter, you want to use microbes like bacteria to recover metals from the waste or the complex material. This eco-friendly technique relies on microbial processes such as bioleaching, using microbes to dissolve metals, or biosorption, where microbes can bind the dissolved metals to produce organic acids or chelating agents that make metals soluble for recovery. So what is the principle of extracting metal from a waste? You have to first leach the metal from the waste. Leach means precipitate it as ions which can dissolve, ions or salts which can be easily soluble or bioabsorbed. So once the metal is soluble in the soluble form and away from the waste complex, then it can be precipitated and the metal can be easily recovered. So copper is one of the most abundant elements in e-wastes or electronic devices like phones and computers and printers and screens. So mostly biohydrometallurgy is used in an electronic waste dumping site where there are such devices, which is then powdered and can be treated via various ways for for biohydrometallurgy. So it can be treated for direct bioleaching in a bioreactor where microbes can dissolve the metals away from the electronic devices, and indirect leaching in a stir tank, while plants can also be used in certain way in certain plants of species can extract or reduce certain metal ions like copper ions in the presence of light in the natural process of photosynthesis, and then these can be purified. These salts and soluble forms of the metal can be purified to aid in metal recovery of these many metals. So copper, gallium, gold, lead, tungsten, everything, all these heavy metals. But copper is one of the most extensively recovered material using biohydrometallurgy. So copper bioleaching is the most common example. Such ores are used, copper ores are used like chalcopyrite, which is cupric ferrous sulfide, or chalcocite or covellite. This is the simplified reaction. So these copper ores or copper containing ores can be used can be used to reduce the ferric ion into the ferrous ion in process oxidizing the copper ion and releasing sulfate. Then bacteria can regenerate this ferric ion by oxidizing ferrous again back to ferric. So this coupling of the copper ore and the bacteria is the form of leaching, in which from the ore the chalcopyrite, cupric or Cu2 plus ions are leached out of the ore. In similar way, in electronic waste, when we have the starting material, in a similar way copper ions can be leached out by using bacteria or microbes, and leach solution contains the Cu2 plus ions, and for such metal recovery then solvent extraction or electrowinning can give us pure copper metal. So any solution from containing Cu2 plus ions, electrospinning can give us pure copper metal deposition, or solvent extraction from the pure solvent is easier. So leaching the mineral ions away from the waste is the job of the bacteria, which facilitates the waste recycling and generations of copper ions. And this is heavily used for regenerating copper from electronic waste. Bioleaching of copper and cobalt, you can see here 100% Cu recovery by biooxidation, in which this sulfur produced is further oxidized, and Fe2 here, cupric Fe2 here, the bacteria involved regenerates the Fe3 plus ions, which performs the attack into the chalcopyrite, or here CuFeS2. So more amount of Fe3 plus produced by the oxidizing bacteria will now attack indirectly to this ore and lead to its leaching, leads to the copper ion leaching. So this is the whole theme or concept of copper ion leaching by biohydrometallurgy. And then the solution which is enriched in copper ion, the copper can be easily extracted from it. Is it clear to the students? If yes, just tell me in the chat box and we can move ahead to the next question. Very good. Thanks, Ankita. Okay. Moving on to the second question. Question number 2. Which of the following elements can be used as the electron carrier during microbe mediated indirect metal oxidation? This is also an easy question. If you have followed the previous question, you will be able to answer this. Follow up of the first question. Which of the following elements can be used as the electron carrier during microbe mediated indirect metal oxidation? Option A, hydrogen. Option B, nitrogen. Option C, carbon. Option D, iron. Or option E, sulfur. Understood the question? Let's take a minute or two to collect some answers in the chat box or in the comment box, and I will revisit this question to reveal what is the correct answer to this question. Okay? Now post your answers in the chat box quickly. Okay, I think that is enough time for this question. Which of the following elements can be used as the electron carrier during microbe mediated indirect metal oxidation? Okay, let us check if people have answered in the chat and comment box. Ankita has answered option D. Okay, now let's see what is the correct answer to this question. Which metal is the electron carrier for microbe mediated indirect metal oxidation? Correct answer is option D, iron. So just as we saw in the last example for biohydrometallurgy and bioleaching, iron can easily serve as an electron carrier, shuttling between Fe3 plus to Fe2 plus states, thus making it a suitable element for electron carrier during indirect metal oxidation. Ankita has answered the correct one. Option D is the correct answer. So what happens in indirect bioleaching or non-contact bioleaching? Iron acts as a crucial electron carrier, repeatedly cycling between ferric and ferrous states. This is what we just saw in the previous example as well, previous question. This process is carried out by specialized microbes which produce a chemical Liggs event that chemically oxidizes the target metal in the ore rather than directly attacking the mineral. So here is the microbe, an example, Acidithiobacillus ferrooxidans. So this can reduce the Fe3 plus to Fe2 plus. So Fe2SO4 3, which is the Fe3 plus state, FeSO4 here. So I'm sorry. This can oxidize back the Fe3 plus population, which can then attack the CuFeS2, which is the chalcopyrite here, from where we want to leach our copper ion. And in the process, Fe3 plus get reduced to Fe2 plus, which is FeSO4. Now this FeSO4 can be consumed again due to this bacteria, Acidithiobacillus ferroxidans, converted back into Fe3 plus. So this acts as an indirect leaching method in which Fe3 plus attacks the CuFeS2, leaching out the Cu2 plus ions into the solution. This process involves two main steps that form a catalytic loop, facilitated by the acidophilic iron oxidizing bacteria Acidithiobacillus. Oxidation of the ore by ferric ion. So Fe3 plus is necessary for oxidation of the ore. In an acidic solution, powerful oxidizing agent ferric ion strips the electron from the metal sulfide ore as shown in the figure, and pH is 1.5 noted, causing the target metal and sulfur to dissolve. In this reaction, the ferric ion is reduced to ferrous ion. Okay, and therefore leaching the metal ion into the solution. Now regeneration of the ferric ion is what is done by the bacteria. The bacteria then use oxygen to reoxidize the resulting ferrous ion back into the ferric ion Fe3 plus, completing the cycle. This step is a primary metabolic function for these microbes which draw energy from this oxidation. Okay, oxidation of Fe2 plus to Fe3 plus, serving as the electron carrier in the electron and fires the electron transport chain and provides energy to the bacteria. So 4Fe2 plus, which is aqueous, plus oxygen in the presence of acid gives rise to 4Fe3 plus 2H2O. So this Fe3 plus, 4 more Fe3 plus molecules are ready to attack some more ores of the CuFeS2, some more molecules of CuFeS2, leaching more metal ions back into the solution. So here I have presented a comparison between direct bioleaching and indirect bioleaching. So what we understood is the method known as indirect bioleaching. Now what is direct bioleaching? The microbes can directly oxidize the mineral, no need for ferric ion intermediate. While in indirect bioleaching, microbes regenerate oxidants which can now attack the ore. Microbe mineral contact is required in direct bioleaching. Via indirect bioleaching, the contact is not required, but the microbe needs to be in the vicinity of the material so that an acidic environment is maintained in which the microbe functions, and also which acts as the oxidizing power from ferrous to generate ferrous to ferric ions. For example, A ferroxidants oxidizing chalcopyrite on the surface, while Fe3 plus ions produced by A ferroxidants dissolve the chalcopyrite. So the same bacteria can perform direct bioleaching as well as indirect bioleaching. Industrial use of the direct bioleaching is while most commonly indirect bioleaching is used, such as Fe3 plus mediated oxidation is faster and scalable. Therefore, indirect bioleaching is the preferred industrial leaching method for collecting copper ions. So any cooperative leaching involves the cell, which is here the ferroxidant cell, which can regenerate Fe3 plus, Fe2 plus to 3 plus, so that now the Fe3 plus can initiate the attack onto the metal surface, onto the ore surface, in turn getting reduced to Fe2 plus, which can now be regenerated by the surface. Whereas in direct leaching, there are other oxidants like SO4 2 minus sulfates and Fe3 plus, which directly attack the ore mineral. Ferrous and sulfur oxidizing bacteria are often used in the direct leaching. So I hope it's clear to the students that iron acts as the metal which can carry electrons and facilitate indirect leaching in process. Yes, sir. Okay. Thank you, Ankita. I hope it is clear to the students. And now let us move to the next question. Question number 3 is on your screen. Which one of the following options presents the correct sequence of event leading to eutrophication? Understood the question? Current sequence of event leading to eutrophication. You have to tell me. Option A, organic matter decomposition happens first, then happens oxygen depletion, then nitrite production, and phosphate solubilization. So from left to right is the order in which the events happen. Option B, organic matter decomposition, phosphate solubilization, oxygen depletion, nitrite production. Option C, phosphate solubilization, oxygen depletion, nitrite production, organic matter decomposition. And option D, phosphate solubilization, nitrate production, oxygen depletion, and organic matter decomposition. While the last option, organic matter decomposition, phosphate solubilization, nitrite production, and oxygen depletion. So, four events, you just have to arrange them in the correct sequence which leads to eutrophication. Understood the question? Okay, let me take a minute or so for allowing all the students and the participants to answer this question. Think about it, remember, recall what eutrophication is and what is the correct order of steps that is involved. Okay, I'll come back later and reveal what is the correct answer to this question. Okay, I think that is enough time for this question. Question number 3. Which one of the following options presents the correct sequence of events leading to eutrophication? Okay, let us check what are the answers posted in the chat box. Tuhin has answered option D. Okay, let us check what is the correct answer to this question. Correct answer is option A. Option A is the correct answer. Organic matter decomposition, oxygen depletion, nitrite production, phosphate solubilization. This is the correct answer. Sorry Tuhin, but option A is the correct answer. Now let us understand what is eutrophication and what are the events that lead to eutrophication. Okay? Eutrophication is the enrichment of water body with nutrients, mainly nitrogen and phosphorus, leading to excessive growth of algae and aquatic plants. As a result, there are algal blooms, high organic matter load, increased microbial decomposition, depletion of dissolved oxygen, death of fish and other aquatic organisms, and decline in the water quality. So, this is what is eutrophication. Due to enrichment of the nutrients in the water body, there is excessive growth of algae and aquatic plants, which blocks the sunlight, which reduces photosynthesis and the oxygen content inside the water. So, depletion of the dissolved oxygen, leading to death of animals and other plants, organic death. Organic matter is organic matter is high in the water. Then the microbes present in the water also start decomposing these organic matter, thereby consuming oxygen, creating a depleted oxygen environment inside, due to which there is death of fish and other aquatic organisms, and the water quality is reduced. There are other microbes which grow, which release lot of toxins. So algae and cyanobacteria are characteristics of the algal blooms, green thick mats of algae and cyanobacteria growing on the top of the lakes. So there is loss of food, habitat and oxygen production inside the lake or the pond in which eutrophication happens. So what is the correct sequence of events? First, there is nutrient load, which can be due to organic matter input and decomposition. From sewage, agricultural waste, algal remains, microbes decompose and increase the biochemical oxygen demand. Then there is oxygen depletion. So nutrient load, algae bloom. Then there is oxygen depletion, nitrogen transformations under low oxygen. So nitrification occurs when there is a low oxygen atmosphere or anaerobic atmosphere. Incomplete nitrification or denitrification, nitrite is formed and reduced nitrogen compounds accumulate in the lake. Then occurs phosphate solubilization. Under anoxic conditions, iron phosphate complexes in the sediments get reduced, releasing phosphate back into the water, which is the internal loading, which is causing more nutrient load onto the water, additional to the phosphate that is received in the organic matter input. This further feeds algal blooms, which is a positive feedback loop. So for our question, this is the step for the eutrophication. First, there is organic matter decomposition. Organic matter decomposition. Organic matter is dissolved in the water, which is first decomposed by the bacteria or the microbes, leading to oxygen depletion. Now low oxygen depletion, low oxygen environments facilitate nitrite production, and after nitrite production there is also phosphate solubilization from the minerals or the sediments present, giving a feedback loop to creating more algal blooms. Well, option D can be very similar or little misleading. Phosphate solubilization, not just phosphate but nitrogen and phosphorus, both are the minerals that are enriching in the water. Then nitrate production is the key wrong point. Then immediately there is no nitrite production. There should be first oxygen depletion due to organic matter decomposition. Organic matter decomposition will produce oxygen depletion because microbes will use up oxygen to decompose the organic matter, then leading to nitrite production. That is why option D is false, but option A is the correct answer. This is the sequence that I will reiterate again. Coming nutrient load in the water, there is bloom of algae and cyanobacteria, leading to which is decomposed. So blooming of algae blocks sunlight, and then algae starts dying and decomposing. This decomposition is brought out by consumption of oxygen. So oxygen depletion leads to death of aquatic life and plants, leading to more biomass which needs decomposition, creating low oxygen environments. Low oxygen environments promote nitrogen transformation into nitrite, and then low oxygen conditions also facilitate phosphate solubilization from the minerals and the sediments, feeding back again to the algal blooms. I hope it is clear to the students. This is the sequence of eutrophication, sequence of events leading to eutrophication. I hope it's clear to the students. Just let me know in the chat box if you have understood, and then we can move on to the next question. Very good. Very good. Prompt response. Okay. Let's see what is the fourth question. Fourth question: which of the following is not a major member of EBPR microbial community? Okay, different set of question. Option A, denitrifiers. Option B, microthrix. Option C, nitrosomonas. Option D, streptococcus. Option E, accumulibacter. The question is, which of the following is not a major member of the EBPR microbial community? So to answer this, you first have to recall what is EBPR and what kind of microbes are a part of this community. Then you just have to find out which of these is not a major member. Easy, right? Well, might not be. You first have to recall what is EBPR. Okay, so let us give some time to this question and come back to look at what is the correct answer. Okay, I think it's enough time for this question. Which of the following is not a major member of EBPR microbial community? Okay, let us check the chat box if people have answered. Okay, so there are a variety of answers. Ashwini says option D. Ankita is saying option C, and Tuhin is saying option D. Okay, let us see what is the correct answer to this question number 4. Not a major member of EBPR microbial community is option C, Nitrosomonas. Nitrosomonas is the correct answer, not a major member. Although Streptococcus also seems to be a correct answer. So I would say this question is a little bit dicey, can have two correct answers. Because Streptococcus is also not a part of EBPR microbial community. But definitely not Nitrosomonas. Because Nitrosomonas is involved in the nitrogen cycle as a nitrifying bacteria. Okay, let us understand what is EBPR and which microbes belong to this community. EBPR microbes are specialized bacteria known as PAOs or polyphosphate accumulating organisms. Now, what is their characteristic? They can take up phosphorus in excess of their metabolic requirements and store it inside the cell as an intracellular polyphosphate granules. So, which we call poly P bodies or P bodies, in which they can accumulate the phosphorus from the environment. They can take up the phosphorus and store them in these granules, and such bacteria are known as polyphosphate accumulating organisms or PAOs. And these bacteria are the ones that are used for EBPR, which stands for enhanced biophosphate recovery. These microbes are exploited in wastewater treatment plants to remove phosphorus and prevent eutrophication biologically. So since nitrogen and phosphorus solubilization is such which important step which increases the organic load inside the water body, such microbes are mostly exploited. The polyphosphate accumulating organism can actually accumulate those phosphates inside their P bodies or phosphate granules, and thereby decreasing the nutrient load on the water body. Common PAOs are Candidatus accumulibacter phosphatis, some Pseudomonas acetinobacter and Microlunatus species also are PAOs. So accumulibacter was one of our options, so it's a big no. Pseudomonas species are also some polyphosphate accumulating organisms. So accumulibacter, the main PAO which is used in EBPR systems. So this is how a polyphosphate body looks like. Phosphorus accumulating organisms are microbes that hyper accumulate phosphorus inside themselves. You can see highly dense condensation of these phosphorus in excess condense into this P body or polyphosphate. In aerobic conditions, these also store volatile fatty acids. So denitrifiers present in anoxic EBPR help in nitrogen removal along with phosphorus uptake. So denitrifiers are another good agents for PAO. They not only help in denitrification or removal of nitrogen but also acts as phosphate accumulating organisms. So it will accumulate phosphate as well as remove nitrogen, thereby decreasing the two main culprits of eutrophication, right? Nutrient load due to nitrogen enrichment and phosphate is what causes eutrophication. Microthrix parvicella is a filamentous bacterium enriched in the EPBR sludge. So it is one of the part of the EPBR sludge, and Streptococcus detected sometimes but not a dominant EBPR functional group. So Streptococcus is ruled out just because sometimes it may be detected, but Nitrosomonas is the correct answer, which is a nitrifying bacteria but not an EBPR. So this is the mechanism of EBPR, which is enhanced biological phosphorus removal wastewater treatment process that uses these PAOs to remove excess phosphate from the wastewater. So first phase is the anaerobic phase where there is no oxygen. These PAOs take up volatile fatty acids like acetate or propionate which is mentioned here, taking up acetate. To do this, they hydrolyze the stored polyphosphate, release the orthophosphate into the medium, and energy in the form of ATP and reducing power from the polyphosphate and glycogen breakdown are used to store these volatile fatty acids as polyhydroxyalkanoids. So this is what happens in the anaerobic conditions. The energy from the reducing power of the polyphosphate and the glycogen breakdown is stored as PHAs, polyhydroxyalkanoids. So as a result, phosphate is released into the wastewater with the uptake of acetate in the anaerobic phase. Then comes the aerobic phase treatment where oxygen is present. In the aerobic phase, these PAOs oxidize the stored PHA for growth and energy generated is used to take up large amount of phosphate from the wastewater to store it back as polyphosphate granules inside the cell. Understood? So these PHA can then be utilized for generating energy and thus also for taking up large amount of phosphate from the wastewater. So net effect, more phosphate taken up than released earlier, right? Because this is in the aerobic process, more phosphate is taken up than released earlier. And waste activated sludge removal, biomass which is rich in polyphosphate storing PAOs is removed from the system. Along with it, phosphorus is physically taken out of the wastewater cycle. So these combined process of aerobic, anaerobic phases and sludge removal is a part of EBPR, which is enhanced biological phosphorus removal. So if you check the levels of PHA, glycogen and phosphate, in the anaerobic chamber we have high PHA because acetate taken up, polyphosphate releasing phosphate, so there is low amount of phosphate. Glycogen releasing energy, breakdown of glycogen in the TCA cycle, releasing energy to store as polyhydroxyalkanoids. Thereby this level is high and acetate levels go down as the bacteria consumes acetate. But what happens in the aerobic tank? In the aerobic tank, the polyhydroxyalkanoid in the presence of oxygen is utilized to give rise to energy, which is fueling the cell's biomass and growth, and also storage of glycogen. Thus intake of phosphate to incorporate into the poly-P bodies. So glycogen levels go up while PHA levels go down and phosphate levels also go down inside the microbe. And then this is activated sludge is cleared, which removes the phosphate together. So this is the EVPR process, and we just saw that Accumulibacter, denitrifiers, microthrix and streptococcus can be grouped into sometimes into polyphosphate accumulating organism, but not Nitrosomonas, which is strictly used for nitrification process. I hope it's clear to everyone what is EVPR, how these organisms, how these microbes are utilized for their potential, and what happens in the aerobic and the anaerobic chambers in an EVPR treatment plant. If it's clear, then let me know and we can move ahead to the next question. Very good. Okay. So moving ahead, question number 5 on the screen. The conversion of ammonia to nitrate is known as dash. This is an easy question now after 4 questions. 5th question is an easy one. Conversion of ammonia to nitrate is known as dash. Option A, denitritation. Option B, denitrification. Option C, nitritation. Option D, nitratation. Or E, nitrification. Ammonia to nitrate is the question. Let me know what you think is the correct answer, and I will wait for a minute or 2 minutes, then come back to this question to reveal what is the correct answer. Okay, post your answers in the chat box and in the comment box. Conversion of ammonia to nitrate is known as what? I think that's enough time for question number 5. Oh, and I see already all three of the students are unanimously saying E. Okay. Conversion of ammonia to nitrate is nitrification. Correct answer. All three of you, Tuhin, Ashwini, and Ankita. Very good. Nitrification is a two-step process. First, conversion of ammonia to nitrite, which is, and then nitrite is then converted to nitrate. So NO2 minus to NO3 minus. This is a two-step oxidation process, two-step microbial process in the nitrogen cycle where ammonia is oxidized to nitrite and then to nitrate with distinct bacteria performing each step. So Nitrosomonas species performs ammonia to nitrite conversion, and the enzyme here mentioned is ammonium monoxygenase or AMO, and Nitrobacter performs nitrite to nitrite oxidation, nitrite oxidoreductase. This aerobic process is crucial for converting ammonia, which is unusable by the plants, into nitrate, which can be absorbed readily by the plants, thus facilitating the nitrogen cycle. So this is the overall nitrification reaction, NH4 plus to NO2 minus to NO3 minus. The process is carried out by special autotrophic bacteria and archaea, which derive energy from inorganic compounds like ammonia and nitrite. Aerobic nitrification requires oxygen and thus is an aerobic process because oxidation reaction, of course, ammonium getting oxidized to nitrite and then nitrate. And it is also a critical step in the nitrogen cycle, making nitrogen available for plants and organisms. The plants can absorb and incorporate it into their building block molecules like proteins. Factors affecting the nitrification. Key factors influencing the rate and efficiency include temperature, level of dissolved oxygen, and the pH and the alkalinity of the environment in which Nitrosomonas and Nitrobacter species can function optimally. So for your reference, this is a nitrogen cycle. How nitrogen is fixed. Atmosphere nitrogen is fixed by nitrogen fixing bacteria, which is fixed in the form of ammonia. Then this is the nitrification reaction we were talking about. Ammonia is converted to nitrite, and then nitrite is oxidized further to nitrates, which can be assimilated inside the plant, and nitrate can be acted upon by denitrifying bacteria to release the nitrogen back into the atmosphere. The nitrate, the ammonia from the organic waste, the nitrogen from the organic waste and dead and decaying matter can be fixed in as ammonia, which then replenishes further nitrite pools. Nitrite can then further be acted upon by nitrifying bacteria to lead increase in nitrate pools, which can then be assimilated by the plants. The nitrogen cycle is the process by which nitrogen circulates between the atmosphere, soil, living organisms and the water. Key steps include nitrogen fixation in the presence of lightning, converting atmospheric nitrogen into ammonia, and then nitrification, converting the ammonia to nitrites and nitrates, and assimilation by the plants, ammonification, decomposition of organic matter, and denitrification, returning the nitrogen gas to the atmosphere. This cycle is essential because enabling the creation of proteins and nucleic acids, but human activities like fertilizer use can disrupt it. So this is the nitrogen cycle, an essential part of which is nitrification reaction of conversion of ammonia to nitrite and nitrate. I hope it is clear to the students, since students have answered correctly. If you want me to repeat it again, please tell me to do so. I'll happily do it. And if you are okay or if you have understood, then we can move ahead to the next question. Please let me know in the chat box. The nitrification reaction, ammonia to nitrite, NO2 minus, and then further oxidation to NO3 minus. I think we should move to the next question, question number 6. Thanks, Ankita. Question number 6. Overall process of nitrogen removal involves the conversion of dash. Again, a tricky question. The overall process of nitrogen removal involves the conversion of dash option A ammonia to nitrite, option B ammonia to nitrate, option C ammonia to nitrogen, option D nitrogen to ammonia, option E nitrogen to nitrate. What is the purpose of overall nitrogen removal from wastewater, and what is the conversion that we are aiming at? Understood the question? Let's take a minute to find out what is the correct answer to this question. Post your answers in the chat box and in the comment box of the YouTube section. I will revisit this question in a minute or two and then let you know what is the correct answer. Okay, I think it's enough time for this question. Question number 6. Overall process of nitrogen removal involves the conversion of dash. Let us check the chat box. comment section. Ashwini says C, Ankita and Tuhin also C. Tuhin has given an explanation also. Very nice. Generally, nitrate to nitrogen, then nitric oxide, and then N2O. Okay. So C is the correct answer. Ammonia to nitrogen conversion. Very well done. You guys have answered correctly. So proud. Okay, let us understand what happens in wastewater nitrogen removal process. So what is the level or what is the current status of the wastewater? When the wastewater is being fed into a treatment plant, there is high level of ammonia, high biochemical oxygen demand, which then has to undergo first denitrification. Denitrification to N2, and then after denitrification chamber, ammonia levels are low, then undergoes nitrification and photosynthetic oxygen, in which these species are generated, which is nitrate and nitrite, ammonia to nitrite and nitrate, which is then fed back into the denitrification chamber, the remnant ammonia, so that these can be then again treated in the denitrification chamber. The nitrates can be converted to the nitrogen, and the flocculum is then processed into the denitrification chamber for the activated sludge treatment of bio nitrogen waste removal. So nitrogen is primarily removed from wastewater through biological process involving two main steps: nitrification and denitrification. The overall goal is to convert nitrogen, mainly in the form of ammonia and nitrates, into harmless nitrogen gas. So then again, Tuhin has explained very correctly. Converting the dissolved ammonia and nitrates to nitrogen is the goal. So converting all the ammonia into nitrates first through the nitrite reaction in the nitrification process, which is then fed back into the denitrification chamber, where it contains denitrifying bacteria converting NO3 minus to N2. Bacteria carry out nitrification process in two steps. All the ammonium salts are first treated by nitrosomonas to give us the N2O, nitrites, which is then treated by the nitrobacter to give us nitrates. And then nitrates can be converted to nitrogen in the presence of denitrifying bacteria. Different type of bacteria, nitrosomonas and nitrobacter. Attributes of nitrifying bacteria: low cell yield, slow growth rate or regeneration time, non-flocculating or weakly flocculating morphology, and highly sensitive to environmental conditions in the reactor. So this is just an information table for the process of conversion of ammonia into nitrogen, which is primarily involved in waste removal of nitrogen from water. I hope this is clear to the students. If yes, just let me know in the chat box and we will move ahead to the next question. Well done for answering the correct answer on the sixth question. Okay, let's move ahead to the next question, question number 7. Thanks, Suhin. Question number 7. Ammonia oxidizing bacteria are preferred in modern biological nitrogen removal systems. Which of the following statement is false about such a system? Understood the question? AOBs or ammonia oxidizing bacteria are preferred in modern biological nitrogen removal system. Which of these is false about such a system? Option A, they require less organic carbon. Option B, they require less oxygen. Option C, they produce less sludge. Option D, they produce more nitrate. Option E, they require inhibition of nitrite oxidizing bacteria. Which of these is a false statement? They require less organic carbon. The ammonia oxidizing bacteria require less oxygen, produce less sludge, produce more nitrate, require inhibition of NOBs or nitrite oxidizing bacteria. Okay, let's wait for a minute and ask all the students to post their answers in the chat box. And I will come back and tell you what is the correct answer. Okay, I think it's enough time for this question. Let us revisit this question and find out what is the correct answer. ammonia oxidizing bacteria, and which of these statements for ammonia oxidizing bacteria in nitrogen removal systems is false? So this is the question. Let us see if people have answered. Ashwini has answered option B and Tuhin is saying option A. Oxidizing bacteria require oxygen but don't know about organic carbon content. Right? Yeah, Tuhin, you are true. You are actually correct. But here the question is asking about which of these is the false statement. Let us understand what is the correct answer to this question. Question number 7. Option D is the false statement. They produce more nitrate. Ammonia oxidizing bacteria does not produce nitrate at all. They oxidize ammonia into nitrites. Rest all the options A, B, C, E are true for ammonia oxidizing bacteria. A or B require less organic carbon, they also require less oxygen, they can produce less sludge, and also they need inhibition of nitrite oxidizing bacteria to function properly. So modern nitrogen removal like partial nitritation NMOX relies on ammonia oxidizing bacteria converting ammonia to nitrite. NMOX bacteria converting ammonia plus nitrite to nitrogen gas directly. This process avoids full nitrification to nitrate and saves oxygen and carbon. So understood? This is the key point here. So AOB and NMOX are coupled together in a flux. So ammonia oxidizing bacteria AOB will convert ammonia to nitrite, which is NO2. And NO2 need not undergo full nitrification to nitrate, which is NO3 minus. It can directly be fed onto the NMOX bacteria, which can convert ammonia plus nitrite to N2 gas. This process avoids full nitrification and saves oxygen and carbon. That's why they require less oxygen and they require less carbon. It also requires suppression of nitrite oxidizing bacteria to prevent the conversion of nitrite to nitrate. Otherwise, NMOX bacteria will not be able to use this as a substrate. Understood? So suppression of NOBs is one reason. Less amount of oxygen and carbon is the another reason. Ammonia oxidizing bacteria are autotrophic bacteria that gain energy by converting ammonia to nitrite, NO2 minus, the first step of nitrification. They are aerobic, require low oxygen levels to survive, and fix carbon dioxide as their primary carbon source. Key characteristics include ability to degrade micropollutants, low growth rates and limited energy gain, presence in various aerobic environments like soil and wastewater. And what happens in ammonia oxidation? AOB performs the aerobic oxidation of ammonia to nitrite, generating energy and fixing carbon dioxide in the process. They are classified as obligate autotrophs, meaning they can only use inorganic carbons like carbon dioxide and cannot utilize organic carbon sources like glucose, and they have low growth yields. So they require less organic carbon is true because NMOX process is autotrophic. No organic carbon is needed. They require less oxygen because only half of the ammonia is oxidized to nitrite and full nitrification reaction is not followed. They also produce less sludge because slow growing autotrophs, low biomass yield. They produce more nitrate. This is false because nitrate formation is undesired and minimal since NOB are inhibited in this case. And they require inhibition of NOBs, which is option E. That is why correct answer is option D, which is the only false statement about ammonia oxidizing bacteria. Is it clear to the students? If you have any doubt or you want me to repeat it, please let me know. Ammonia oxidizing bacteria coupled with NMOX is the modern biological nitrogen removal where there is no full conversion of nitrite to nitrate. Rather, NMOX bacteria can directly convert nitrite pools with ammonia to nitrogen gas. Low yielding, low energy, low oxygen requirement and requires inhibition of NOBs. Very good. Thanks to him. Let us move to the next question. Question number 8. Endamo archaea archaea offer a unique advantage in biological nitrogen removal by virtue of their ability to use dash as an electron donor. N-Damo archaea or N-DAMO archaea have the advantage because they can use which of these as an electron donor? Option A, ammonia. Option B, nitrite. Option C, hydrogen. Option D, methane. Option E, methanol. Understood the question? Biological nitrogen removal. The bacteria in question is N-Damo archaea. Sorry, it's not a bacteria, it's an archaea, which can use dash as an electron donor. Okay? Please tell me what do you think is the correct answer. Answer in the chat box, please. I will revisit this question in a minute or two and tell you what is the correct answer. Okay, let us see what is the correct answer to this question. What have people answered in the chat box? Tuhin says option D, Ankita says option D, Ashwini is answering option A. Okay, Tuhin has an explanation. Archaea are methanogenic microorganisms, but ammonia can be used as electron donor. Okay, we will come to the explanation after finding out what is the correct answer to this question. Question number 8, N-Damo-Archaea. Correct answer is option D, methane. So Tuhin and Ankita have answered correctly. Sorry Ashwini, the answer is option D, methane. So methane is the electron donor in N-Damo-Archaea, which offers advantages in biological nitrogen removal. Now let us see what kind of advantage and what is N-Damo. N-Damo, or the short form NDMO, stands for nitrite or nitrate dependent anaerobic methane oxidation bacteria. It's a microbial process where methane is oxidized without oxygen using nitrite or nitrate as the electron acceptor. So you have to be careful here. Methane is the electron donor while nitrite or nitrate is the electron acceptor. So how does nitrate reduction happens? By this archaea, methanoperidine oxidizes methane using nitrate as an electron acceptor. CH4 plus 4NO3 minus gives CO2 plus 4NO2 minus plus 2H2O. And by other bacteria, which is not an archaea, methylomirabilis oxifera takes up produced nitrite and converts it into nitrogen. But interestingly, it generates intracellular oxygen from the nitric oxide. Like 2NO2 minus gives 2NO, and then 2NO gives N2 plus O2. Thus, the O2 formed is used to oxidize methane inside the same cell. So this is a methanogenic bacteria, while this is a methanogenic archaea. So methane is the electron donor here, and the nitrate is the electron acceptor. Methane donates electron, nitrate is reduced to the nitrite. Now what happens when Damo bacteria, N-Damo bacteria is coupled with Anamox? So we just understood this reaction, methane as the electron donor reacting with nitrite to give rise to reacting with nitrate or nitrite and reducing it, removal of oxygen from the nitrite to produce nitrogen. Electron donor is methane and electron acceptor is the nitrite or nitrate. These are the archaea, methanoperiden's nitroreducens. It links the carbon and nitrogen cycles, enables methane removal and denitrification simultaneously, and operates in oxygen limited zones of wastewater and sediments. So these are some of the advantages N-Damo bacteria provides. So methane conversion to CO2 as a result can utilize NO3 minus or NO2 minus as electron acceptors in the Anamox chain, where more amount of ammonia can be consumed for producing NO2 minus and NO3 minus. Understood? Since the Damo is confusing, since Damo is using up all the nitrite as an electron acceptor and converted it into atmospheric nitrogen, more amount of ammonia has to be pushed into the reaction so that more amount of nitrite and nitrates are generated, thus enabling nitrogen removal, methane removal and denitrification simultaneously, linking the carbon and nitrogen cycle. So thus NDMO, in the NDMO bacteria or archaea, methane is the electron donor while nitrite is the electron acceptor. I hope it is clear to the students. This is the diagram which will help you clear things out. That in NDMO archaea, the conversion of methane to carbon dioxide happens by consuming nitrites and nitrates, which are the products of Anamox reaction, which are the products of ammonia oxidization. Ammonia is oxidized to nitrite and nitrate, which is consumed by the DMO bacteria, thus putting more pressure on this reaction to generate more of these. So more amount of ammonia is removed from the wastewater, while methane is also removed. Methane removal is also done and denitrification is done simultaneously, and it can operate in low oxygen zones and wastewater and sediments. Okay, thanks Ashwini. Moving on to the ninth question. Which of the following microbial products is correctly matched with its application in oil recovery? Okay, so we have reaching to the final couple of questions in this live TA session. Now the topic we are dealing with is oil recovery. Which of the following microbial products correctly matched with its application in oil recovery? Option A, biomass oil dissolution. Option B, biopolymers rock wettability alteration. Option C, solvents viscosity reduction. Option D, acids oil degradation. Option E, gases oil emulsification. Which of these is a correct match? Okay, that's enough time for question number 9. Let's check what are the answers in the chat box. Which of the following microbial product is correctly matched with its application in oil recovery? Ashwini says option C. Ankita option C. Tuhin says option C. Oh, he's changing his answer to B. Okay, let us see what is the correct answer to this question. Option C is the correct answer. Solvents are used for viscosity reduction. Tuhin, C was the correct answer. I'm sorry if you got confused, but Ashwini and Ankita have answered correctly. Now let us understand what all products are required in oil recovery and what is the process of oil recovery. Microbially enhanced oil recovery, MEOR technique, is the one used for recovering oil from the sediments. Involves injecting specific nutrient rich substrates like bacteria and biosurfactants, encouraging indigenous and specially introduced microbes to flourish. And these microbes metabolize, they produce various products that work in concert to reduce the viscous forces, binding of the oil to the rock, increase the forces that push the oil out of the rock pores, and improve the flow pathways within the reservoir, ensuring more efficient sweeping of the oil. And then these oil are swept towards the production well, which can then extract the oil from the water in immiscible forms. So first in the injection well is what is loaded with the bacteria, nutrients and biosurfactants, pressing the water containing microorganisms, biosurfactants and nutrients, enhanced mobility and biodegradation of crude oil to low molecular weight. Microbial metabolites are generated like gas, acid, biomass and polymer. Crude oil is swept away from the injection well towards the production well. Improvement of the crude oil mobility, improvement of oil reservoir percolation and enhanced oil recovery, and advanced water is pulled up from the production well. Now in microbially enhanced oil recovery or MEOR, different microbial products act through distinct mechanisms. So biomass oil dissolution, not correct because microbial biomass mainly helps in selective plugging of high permeability zones to redirect water flow and not oil dissolution. So more of the biomass will plug so that water flow can be unidirectional towards the collection area or the production well. Biopolymers, rock wettability alteration. Biopolymers like xanthan gum are used in the injection well to increase the water viscosity and improve sweep efficiency, not alter wettability. So improving the sweeping efficiency of the oil and water towards the production well. So wettability is mainly influenced by biosurfactants and not biopolymers. So you need to understand that the hydrophobic oil and the water, when we have wettability of a hydrophobic substance, we always have the involvement of biosurfactants or detergents which can enable or reduce the surface tension between the hydrophobic molecule and the water. Solvents reduce the viscosity. Microbial solvents such as ethanol or butanol produced by the Clostridium, which is one of the MEOR, reduce oil viscosity and dissolve heavy oil components to improve the oil flow. Hence, this option is correct. Acids in oil degradation, incorrect because organic acids help in rock dissolution, improving porosity and permeability, but they do not degrade oil significantly. So, since the sediment and the rocks are the ones which is housing the oil, mostly acids act on the rock and rather the oil. Gases oil emulsification, again emulsification is mainly caused by biosurfactants. So emulsifiers are biosurfactants. Gases like CO2, methane increase the reservoir pressure and helping displace the oil and not emulsify it. Emulsification is mainly caused by surfactants. So therefore C is the only correct option where solvents are used for viscosity reduction. All other options are incorrect matches. Understood? Is this clear to the student? I have analyzed each and every option and what is the property that they assist in the microbially enhanced oil recovery method. Very good. Thanks, Akshwini. Now coming to the last question of this session, again on microbial enhanced oil recovery. The most suitable genus for MEOR is dashed. This is an easy question. Option A, Klebsiella. Option B, Clostridium. Option C, Pseudomonas. Option D, Enterobacter. Option E, Methanobacterium. Which of these is the most suitable genus for MEOR? I think we need not wait much time for this question. I can already see some answers in the chat box. Ankita is saying option B, Tuhin option B, Aswini says option C. Okay. So let us see what is the correct answer. Correct answer to this question is Clostridium. Option B is the correct answer. Sorry, Ashwini, but Pseudomonas is also a good candidate, but Clostridium wins by a large margin because Clostridium produces a variety of these products, bio these products which aid in enhanced oil recovery. So as an all-rounder, Clostridium is the winner here. So option B is the correct. Tuhin and Ankita have the correct answers. Well done. Clostridium is the correct answer. So again, let us revisit the MEOR system. Bacteria and nutrient or biosurfactant is injected into the injection well, which drive the water for enhanced oil mobility, biodegradation of the crude oil. Microbial metabolites like biosurfactants, polymers and acids and gases help in different procedures that we just learnt in the previous answer. Then the crude oil is pushed towards the production well, which is then extracted in enhanced oil recovery. So when most oil has already been extracted by physical and water flooding methods, large fraction of the oil remains trapped in rock pores due to surface tension, viscosity and other factors. So microbial enhanced oil recovery uses microbes to improve oil flow and extraction efficiency in overcoming these physical and chemical barriers like surface tension and viscosity. So injection process has bacteria plus nutrients injected. The outcome is to colonize the microbes inside the reservoir. Metabolism, microbes produce many surfactants and gases which decrease the oil viscosity and increase the mobility and flow of the oil from the rocks. Displacement, gas and metabolites push the oil and enhanced flow to the production well. While production, oil plus water is pumped down and higher oil recovery yield is obtained. So why Clostridium? Because it confers many advantages. Suited for anaerobic reservoir conditions, and deep oil reservoirs are mainly oxygen deficient because they are anaerobic. Clostridium species are strictly anaerobic, meaning they can thrive and remain metabolically active, whilst Pseudomonas is not strictly anaerobic. They grow on cheap carbon sources like molasses, making them cost effective in field application. Another reason. So environment, anaerobic environment, ideal for Clostridium. Metabolites such as carbon dioxide and organic acid, mild surfactants also aid in emulsification, decreasing the oil viscosity and increasing the mobility. Pressure increases the viscosity reduction, interfacial tension lowering, and significant enhancement of oil mobilization and recovery efficiency on cheap cost. Therefore, this is a popular choice of bacteria which are used for MEOR or microbially enhanced oil recovery. Clostridium is the correct answer. Is it clear to the students? If it's clear to everyone, very good, Tuhin. Thank you. So with this, we have come to the end of our class. So in a span of 1.5 hours or 1 hour 45 minutes, we have tried to solve 10 multiple choice question. I will stop my presentation. So in this short period of time, we have tried to solve 10 multiple choice question. We covered important topics like nitrification, nitrogen waste removal, polyphosphate bodies, and EBPR. We also saw ammonia oxidizing bacteria. We covered topics like oil recovery. So I hope everyone was agreeing with me, was trying to answer. Great participation. I liked the class as well. If you have any suggestions on how this class should be operated and carried on, please feel free to drop your suggestions. If you have any suggestions on the slides and the way I conduct classes, then please do so. If you like the class, then well and good. This class is a recorded class, so the recordings will also be available, as well as these questions along with the materials that I have prepared. The presentation will also be provided. So if you have no further doubts, let us meet the next Saturday with week 12 lectures. Same time, 3 p.m. Saturday. We will be solving 10 such questions from lecture week 12. Till then, read your lectures, prepare for your examinations, and I hope you are enjoying these classes. Thank you for joining. I will end this call right now. Thank you.